First of all, for our introduction, Andrographolide is a principal bioactive compound found in Andrographis paniculata, which is well known as the king of bitters due to its intensely bitter taste. This herb has been used traditionally in various systems of medicine worldwide since ages in Ayurveda, Chinese and Malay. It belongs to the Acanthaceae family and has a long history of use in traditional medicine systems such as in traditional Chinese medicine. The plant is an annual herb with lens-shaped leaf and small white flowers. Also, various parts of the plant, especially the leaves and the stems, are used for their medicinal benefits. Now we move to the Andrographolide. This compound has been identified as the key contributor to the plant's medicinal properties. Andrographolide has gained attention for its diverse pharmacological activities, including anti-inflammatory, antiviral, antibacterial, and anti-cancer effects. In recent years, research has focused on exploring the potential of Andrographolide as a therapeutic agent for various health conditions, with a particular emphasis on its anti-cancer properties. Here is given the structure of Andrographolide and its IUPAC name. Next, for potentially treatable disease, Andrographolide AG has been studied for its anti-cancer properties, including anti-proliferative, which has shown promise in slowing down cancer cell growth, anti-angiogenic, which is necessary to block the formation of new blood vessels that feed tumors, and anti-inflammatory activities for reducing inflammation associated with cancer. These properties make Andrographolide an interesting subject for cancer research. Studies also have demonstrated the effectiveness of AG in inhibiting the growth and progression of several cancer types, including breast cancer, prostate cancer, colon cancer, liver cancer, leukemia, and lung cancer. As a treatment for cancer, Andrographolide has been shown to induce cell cycle arrest, apoptosis, which is a programmed cell death process in various cancer cells. This is crucial for preventing the uncontrolled growth of cancer cells. Also, it can inhibit the growth and invasion of cancer cells in the patient's body. Next, chronic inflammation is often associated with cancer development. Andrographolide exhibits strong anti-inflammatory properties by inhibiting pro-inflammatory cytokines and pathways, potentially reducing the inflammatory microenvironment that supports cancer growth. Besides, it can inhibit angiogenesis, the formation of new blood vessels, which is crucial for tumor growth that led to potentially decreasing tumor blood supply and limiting their growth. AG also modulates the immune system, enhancing the body's ability to recognize and eliminate cancer cells, and helps counteract oxidative stress, which is implicated in cancer development. By scavenging free radicals, Andrographolide may protect cells from DNA damage and mutations. Moreover, Andrographolide has shown some degree of selectivity, meaning it focuses more on attacking cancer cells and causing fewer impacts on healthy cells. For the last one, Andrographolide may enhance the effectiveness of conventional cancer therapies, potentially acting synergistically with chemotherapy or radiation treatment.
Now, I'm going to continue the presentation, which is the pharmacokinetics. First of all, what is pharmacokinetics? Pharmacokinetics is the effect on how the body do to the drugs. In order to understand the pharmacokinetics better, you can ask yourself a question, which is how does the drugs get into the body, how does the body do to the drugs, and how does the body eliminate the drugs. Pharmacokinetics consists of four main processes, which is absorption, distribution, metabolism, and also excretion. Now, let's get into the first main process, which is absorption. Andrographolide gets into the body through two ways, which is intravenous administration and also oral administration. For oral administration, Andrographolide will be absorbed through the duodenum and also duodenum. The absorption mechanism is through the passive diffusion, and as we all know, Andrographolide is not a water-soluble. Thus, the Andrographolide will be metabolized into more a water-soluble compound. This will be explained further in the mechanism process. Based on the research paper that I have studied, Andrographolide gets absorbed from the gastrointestinal tract into the blood with an absorption half-life of about 25 minutes. Now I will explain the second main process, which is the distribution. It was found that andrographolide bound extensively to blood proteins and redistributed between blood and tissues. The time taken is between 1 to 2 hours. Based on the article that I have studied, highest concentration of andrographolide is found in kidney, followed by liver, spleen, and the last one is brain. Now is the third main process, which is the metabolism or biotransformation. For the metabolism of andrographolide, glucuronidation and also sulfonation occur in the metabolism of andrographolide. This metabolism of andrographolide is occur in order to increase the bioavailability of andrographolide. First of all is the sulfonation. Sulfonation involves the addition of a sulfur group, which is SO3, to a molecule of andrographolide. Then, andrographolide undergoes sulfonation to produce the identified sulfonate metabolite. This introduces a highly polar and water-soluble functional group, making the modified compound more hydrophilic. When the compound more hydrophilic, this will increase the water solubility of andrographolide. The increased water solubility is crucial for the efficient elimination of substance from the body, primarily through urine. Andrographolide was metabolized to a sulfonate 14-deoxy 12-sulfo-andrographolide. The second metabolic pathway of andrographolide is glucuronidation. Glucuronidation is a phase 2 metabolic reaction in which a glucuronic acid moiety is conjugated to a substrate, forming a glucuronide conjugate. The glucuronide conjugate is a more water-soluble form of andrographolide, making it easier for the body to eliminate this metabolite. Thus, through these two metabolic pathways, andrographolide can be more water-soluble and can be successfully excreted through urine. Now, let's move on to the last main process, which is the excretion. The glucuronide and sulfate derivative were finally eliminated into the urine until 48 hours after oral dosing. More than 90% of the 14-deoxy 12-sulfo-andrographolide was eliminated through other ways, probably through metabolic transformation.
Now, I'm going to present the pharmacodynamics. Andrographolide is a drug that has the cytotoxic action, where a drug that has the ability to induce cell death or damage in living cells. So there are two mechanisms of how andrographolide help in the treatment of cancer. First, cell cycle arrest. So what happened in this cell cycle arrest? First, cyclin-dependent kinase CDK inhibition. The CDK inhibitors competitively inhibit the activity of cyclin-dependent kinases. They bind to the ATP binding site of CDKs, preventing the binding of ATP and substrate molecules. This competition blocks the phosphorylation of target proteins involved in cell cycles progression, leading to cell cycle arrest. Next, p53 activation. Tumor protein p53 is a tumor suppressor, which means that it regulates cell division by keeping cells from growing and dividing too fast or in an uncontrolled way. The second mechanism is apoptosis via mitochondria. What happens in this apoptosis process? First, cellular stress and activation of the PCL2 family proteins. Various cellular stresses, such as DNA damage, lack of growth factors, or oxidative stress, initiate the apoptotic process. Pro-apoptotic members of the BCL2 family, such as BAS and BAK, are activated in response to these stresses. Meanwhile, anti-apoptotic proteins like BCL2 and BCLXL are inactivated. Next, permeabilization of the outer mitochondrial membrane. Activated BAS and BAK earlier promote the permeabilization of the outer mitochondrial membrane. This process is often referred to as mitochondrial outer membrane permeabilization. Permeabilization allows the release of proteins from the intermembrane space of the mitochondria into the cytoplasm. Next, release of pro-apoptotic proteins Key pro-apoptotic proteins released include cytochrome C. Next, formation of the apoptosome. The released cytochrome C earlier will bind to the APAF1, apoptotic protease activating factor 1, to form apoptosome. This complex recruits and activates pro-caspase 9, which is an initiator caspase, through a conformational change and proteolytic processing. Next, activation of caspase cascade. Activated caspase 9, then activate downstream effector caspase, which is caspase 3. The effector caspase in turn cleave a variety cellular substrates, leading to the characteristic features of apoptosis. Next, execution of apoptosis. The activated caspase orchestrates the cellular changes associated with apoptosis. They cleave structural and regulatory proteins in the cytoplasm and nucleus. Lastly, phagocytosis of apoptotic cell. The apoptotic bodies containing cell fragments and organelles are recognized and phagocytosed by memory cells or professional phagocytes. This process prevents the release of potentially harmful cellular contents and ensures the removal of apoptotic cell without including inflammation.
Going to the methodology on extracting and analyzing the purity of drug, which is andrographolide. First, we need to obtain the leaf of Andrographis paniculata plant. After that, it should undergo drying process by air dried under shade for 1 day and dried under hot air oven below 60 degrees Celsius. Next is powdered process, where the leaf is green until powder obtained and kept into 15 to 20 Celsius airtight container. Going to the next procedure, which is extraction and isolation. It is extracted by 3 different solvents. Not only that, the dark green crystalline, which is obtained from the extraction and isolation before, is washed with toluene until most of the coloring matter is removed. Next, product is dissolved in methanol and cooled in refrigerator for further recrystallization. After this, analysis is done to ensure the purity of andrographolide obtained. So I've mentioned before that 3 samples extracted by using 3 different solvents. The first sample is extracted by using 1:1 ratio of dichloromethane and methanol mixture by cold maceration, while the second sample is in ethanol and macerated overnight in 10 cycle. And for the third sample is also macerated overnight in 10 cycle, but it is used different solvent, which is 95% alcohol. After this extraction, the extract is filtered and the solvent is removed under vacuum. Next, going to analysis compound. The analysis to done to ensure compound's purity by using UV, IR, and MS method to analyze and compare spectrum produced with reference standard. The solvent used in this analysis is chloroform, methanol, and also ethyl acetate in ratio 8:1 to 5:1. Result obtained can be seen in UV spectrum where the data match with the reference standard. Thus, 3 samples of andrographolide analyzed is pure andrographolide.
Andrographolide can be identified by looking at its physical characteristics with molecular weight of 350.4 gram per mole and melting point of 229 until 232 degrees Celsius. It can only be stored at 2 until 8 degrees Celsius and it usually in form of white solid. It is also physically stable for 2 years from date of purchase as supplied. From chemical view, it is easy to hydrolysis, open loop and isomerization in the aqueous solution to affect the drug stability. The most stable pH value is 3 until 5. At lower temperature, the stability of andrographolide is better. It is also dissolved in boiling ethanol, slightly soluble in methanol or ethanol, very slightly soluble in chloroform and almost insoluble in water. Andrographolide is more stable in chloroform. For biological characterization, various research have found that this drug is antioxidant, anti-inflammatory, and antibacterial. It is also immunomodulatory and cytotoxic to human at the same time. Not to mention that it is neuroprotective and hepatoprotective.
Last but not least, I will continue with clinical trial, which is in vivo experiment. This experiment is conducted by using BASRI mice. BASRI mice is an albino laboratory-bred strain of the house mouse. The BASRI mice used in this experiment is between 25 until 28 gram and 6 until 8 weeks years old. They also taken from AMALA Research Center breeding sessions and conducted according to Animal Ethics Committee. The actual purpose of the experiment to activate the cytotoxic T lymphocyte, which is CTL, response and attenuation of cancer growth by the Andrographische paniculata extraction, which is APE, and Andrographolide, which is ANDLE. This is because CTL production can inhibit the cancer growth and increase the lifespan of the mice. CTL production was determined in BASRI mice by Wie's neutralization assays using sensitive EL4 thymoma cell as the target cell. Before I go through deeper in this clinical trial, there are some terms that you must know. First, EL4 thymoma cell is a cancer cell that contained in the mice body. Second, lifespan is defined as the period of time between the birth and death of an organism. The increasing in lifespan will increase the survival period of the organism. Lastly, percentage ILS is the percentage of increase in lifespan. Next, moving to the table showing the effect of Andrographische paniculata extraction and Andrographolide on in vivo CTL generation and survival of animals. According to the table, lifespan of untreated EL4 cancer-bearing animal is 27.1 days. But when these animals receive 10 doses of APE and ANDLE, the lifespan increased to 51.1 plus minus 1.9 days and 44.5 plus minus 2.4 days respectively. Besides that, the percentage in increasing the lifespan also increased to 88.5% and 64.2%. The survival time of treated animals by APE and ANDLE increased comparing with that of animals receiving cancer cell EL4 alone. The increase of lifespan of the animal is directly proportional to CTL activity. So, this clinical trial proved that Andrographische paniculata extraction and Andrographolide are very effective to inhibit the cancer growth by stimulating the CTL production.